both. We are delighted to have Professor Michael C. Frank here today to discuss moving towards a more cumulative science in developmental psychology. I'm sure many of you are familiar with Mike's work, but for those of you who are not, Mike is the David and Lucille Packard Professor of Human Biology at Stanford University and received his PhD from MIT in Brain and Cognitive Sciences. In particular, Mike's promoted a sort of group science approach to developmental psychology, and in particular on language use and language learning. So he's the founder of the ManyBabies consortium, with which many of you will be familiar, but also has led open data projects, including Wordbank, which is an amazing resource for developmental researchers, and MetaLab. So I could spend, a great long time sort of blowing Mike's trumpet for him, but I think best if we allow him to jump in and tell us about cumulative science in developmental psychology. Thanks, Mike.
Thanks for having me. Appreciate the invitation and the kind introduction. And I hope to ruin that immediately by just starting by talking about all of my mistakes. And so I'm really excited to be here and to tell you a little bit about some of our efforts on thinking about psychology as a cumulative enterprise. But I'm also serious about the mistakes. So let me start with early mistakes. So this is my senior honors thesis from my undergraduate degree, now more years ago than I expected. I was interested in morphology and morphology learning and second language learning, and I followed lots of contemporary practices that I was trained in, when I was collecting data, I collected data and tested my statistics along the way, checking to see if my p-value was less than 0.05 and only stopped collecting data when it was. Then, at the advice of my advisors, I added an extra condition later, then did statistical analyses directly comparing those conditions. Probably would have added another if that third one hadn't worked. And then, still in the absence of any really strongly significant results, I post-hoc split by a variable that I collected after the fact, found a somewhat surprising pattern, and then interpreted it despite evidence that was above my notional alpha threshold. So I would say this was suboptimal in a number of ways, but it was a very interesting learning experience, and it really got me passionate about working with data, and so it served its pedagogical purpose. Then, during my work as a research assistant in an infancy lab, I started working on this wonderful data set of children's free viewing of movies. And we were interested in a variety of things, really. I should say we weren't interested in their attention to faces initially, but we noticed that they were attending to faces a little bit less in the younger age groups. And so that's what our paper is about, right? We backed into this paper that was about the distribution of attention, and it had some very pretty figures in it that showed increasing attention to faces over the course of development and an increasing tightness of children's fixation. But just in the nature of full disclosure, this is only a very small subset of the beautiful figures that I made. In fact, I used this data set during graduate school to learn every single analytic technique that I possibly could. I tried them all on this data set. Mixture of Gaussians, got it. Kernel density estimation, yep. Probabilistic modeling, Yeah, exploratory visualization of other types. Yeah, we did that. I really just tried the whole gamut, and the ones that looked prettiest and were most interpretable made it into the paper. So then that made the 4-year gap between this data set and the next one more nerve-wracking, as I wondered, did I really, in fact, discover anything at all here, or had I just mined something out of the noise? Well, luckily, eventually we were able to collect another comparable data set, which we analyzed in, I think, slightly more sensible ways, and we found that face looking did indeed increase in our stimulus, and in fact, even to some extent in another one. So luckily, we were able to replicate our initial exploratory finding, but there was some substantial anxiety on my part once I realized that this exploration, this unbounded exploration, and then writing up the results of the exploration as though they were what we were looking for all along was maybe a little bit suboptimal. Finally, here's my third paper, and I hope that I'm really I'm hoping that I'm making you feel good about selecting me as a speaker, since I'm maybe more error prone than or bad practice prone than others that you could have selected. So this paper was an investigation of what's known as rule learning. So it's a generalization of a regularity from some kind of repeated exposure. And then the regularity is generalized to new stimuli. This was a paper, again, where we tried to do this with infants, and we found that multiple modalities of information was important in generalization. The paper went through peer review, 2 supportive rounds of peer review with an expert editor and expert reviewers. This was figure 1. It showed clearly our generalization result, where we got dishabituation to incongruent rules relative to congruent rules. And despite my co-authors and I looking at this fairly extensively, and the reviewers and the editors, nobody really noticed that there was something a little bit odd about our habituation times in this experiment. Our habituation times were vastly lower than our dishabituation times, even in the congruent condition that should have been fairly comparable. In fact, they started about 6 seconds and they went to about half a second. That wasn't what happened. Just spoiler alert. We had to fix that. That was a typo in my code. I was writing some particularly uninterpretable MATLAB code at the time, and I indexed into an array incorrectly. Those are the few babies that continued looking through 6, 7, 8, 9, 10, 11, 12 trials. There's very little data here. It's really basically meaningless. This is what the figure should have looked like. This is a real habituation curve coming down from about 50 seconds to 15 seconds, 10, and so forth. And then it very nicely matches the dishabituation. Note that the axes are right here. So, okay, these are really some of my first research projects, and my gradual awareness of changing practices and better ways of analyzing data and thinking about inferential conclusions and statistics, made me think that there were some things that were a little suboptimal about these practices. But these were just my personal experiences, and I've known throughout my life that I'm a sloppy and error-prone person, unfortunately. So maybe the broader field is actually fine. Of course, as I was discovering these mistakes myself, I was also getting educated about the possibility of these sorts of mistakes by others. And for a while, those were just in the status of anecdotes. So really, you could still think that things are really fine, and there was no need for kind of corrective action at the broader policy or scientific level. But I did start to wonder, what are the foundations of the work that we're trying to build on? And especially as I think of myself as kind of a theorist, I try to build computational theories of children's learning. How effective was that theory building when I was building theory around data points that might have looked a little bit like the ones I was just showing you that I was creating myself in my empirical work? So in this talk, I want to tell you a little bit about issues of reproducibility and replicability, maybe more broadly, and then in developmental psychology. And then I'll talk a little bit about models for cumulative progress. So let's start just with the issues of reproducibility and replicability. As I started in this field of metascience that was emerging, it quickly became clear that one of the most amazing transitions in the, say, early 2000s was the transition between individual anecdotes like the ones I just told about scientific practices going wrong to a more systematic, policy-based investigation of the flaws and issues in the scientific ecosystem. And so the way that kind of policy conversation goes is you need to think about the outcomes that you really want, and then consider measuring progress towards those outcomes and whether particular changes to our scientific policies or ecosystem could improve those outcomes. So here are just a couple of the outcomes that we care about. So if I've got the same data as you do, and I'm running the same code or the same analyses, and I get, for some definition of the same result, let's call that a reproducible finding, computationally or analytically reproducible finding. I'm literally just getting the same numbers that you did. If I'm doing the same analysis, but I'm collecting a new data set, that we could call a replication study, and if that works out, we might call that a replicable finding. On the other hand, if I'm looking at the same data, but I have a different analysis specification, different code, I might call that a robust finding if I find the same thing with different kinds of analyses. And finally, you might think that something that's robust and replicable might be more likely to be generalizable, although, of course, issues of sampling and sample comparison do come into play there as well. So all of these are critical prerequisites of a cumulative science. And these outcomes have been the target now of a variety of meta-scientific investigation. And that meta-scientific perspective is something that's really helped me think about my own errors in context and think about the ways that they might pervasively have an effect on the scientific ecosystem and the literature, and whether we can, in fact, build on it to create cumulative theories that explain, in maybe in a positivistic way, more and more of the data and more of the variation that we see in kids learning. So the critical experience for me here was participating in one of the first big psychology-based open science Metascience projects. This was a project that was coordinated by Brian Nozick. When I started at Stanford, I was working on a course design, and I built on a course that Rebecca Sachs had designed at MIT and that I had TA'd for, where Rebecca had the students do replications of the published literature. In fact, she did replications and extensions, which was very ambitious and a lot of fun. And so I was doing this course on replication, and Brian was doing a sabbatical at Stanford, and he said, hey, you know, in your replication class, why don't you do some replications from this systematic sample that we're going to do? So we're going to do 100 studies. Actually, I think they didn't even know it was going to be 100 at that point. But we're going to do 100 studies of papers from 2008 from top journals, and we're going to see how many of them replicate. And the disappointing result, when all was said and done several years later, was that only about a third of these, by a variety of criteria, in fact, showed something like the original result. So this paper generated a lot of heat and light. A lot of people got interested in it. A lot of people started doing Metascience. Also, a lot of people hated it and thought it was terrible. I think the truth is somewhere in between. This is what I would think of in psychology as a fabulous study one. It's big, it's ambitious, it's different, it's exciting. It also had severe inferential limitations. So the replications weren't necessarily appropriately powered. There weren't always perfect matches between the stimuli of the replication and the stimuli of the original. Hypotheses were maybe chosen to test a bit ad hoc, and so forth. There were, in hindsight, some substantial issues. But the wonderful thing about a new ecosystem, new literature to work in is that you can do not just study one, but studies 2, 3, and 4. And this Metascience field has in fact advanced pretty substantially, and so you get papers just a couple years later, like this one. This is Kammerer et al. 2018. I wasn't involved in this at all, but here the sampling frame is much clearer. They take all of the papers with simple behavioral experiments in the journals Nature and Science between 2010 and 2015. So there's no issue of sample generalizability. This just is the sample. Then they do very highly powered, very high fidelity replications. And then when those don't, if those don't pan out, those are the yellow points in this first graph, they actually add additional participants and consult with the original authors and really try to make these as good as they can be. And here now you start to get a top line replication rate closer to 2/3, well, 60% maybe, depending on how you aggregate the evidence. So now there's quite a cottage industry of these kinds of projects across different literatures, and you can argue about what the optimal outcome is, but it seems like between 50 and 65% replicability is what people tend to find. So some of the worries that I had about my own work and whether it would replicate, in fact, I think were justified in this case, because in general, when exploration or low power studies then get promoted into journal publications, they don't always replicate. But maybe an even more worrisome issue is the second one that I highlighted, the errors in my work, that maybe findings aren't analytically reproducible. And I actually, this is the stuff that radicalizes me, because I think we can wonder about when replication is the correct approach, when generalizability is more important, when boundary conditions are important. But I don't think anybody thinks that a paper should have numbers in it and you don't know where those numbers come from. You always want the numbers in the paper to be right. Even if you can never do that experiment again, even if it's not replicable because it was a unique moment in history, you still want the numbers to be calculated correctly. And they aren't always. So there's this very clever and wonderful research program by Jelte Wijkerts and their collaborators, Michel Nuiten, in the most recent work, where they take APA-reported statistics and they compare the degrees of freedom, the p-value, and the test statistic. Because those pieces of information are redundant with one another, you can figure out if they match up. And if they don't match up, something is wrong, because that's logically impossible. It's mathematically impossible. So you can tell a lower bound on the number of errors in the statistics that are being reported in papers. And that lower bound is actually like upsettingly high, right? In this initial paper, in a smaller sample of statistics, you actually see that about 15% or so of p-values or of inferential statistics are incorrect. And that's really worrisome. In fact, in Michel Nuiten's larger analysis, you see that this lower bound is about half of all published psychology papers have some sort of reporting error in them, which is really quite distressing, actually. It's really upsetting. But that's a lower bound. So how do you actually find out whether papers are really analytically reproducible? Well, that's pretty tricky. This is a project that Tom Hardwick led while he was a postdoc in my lab, or actually a pair of projects. So we took advantage of open data policies at several journals, Cognition for one, and then Psych Science through the Badges program. And as part of that, an evaluation of those programs, we actually downloaded a whole bunch of these data sets, 35 in the Cognition case, 25 in the Psych Science case, and we laboriously re-implemented exactly the analyses that were specified in the paper. This was a huge amount of work. This took 10, 20 hours per project, for each of these papers. It's like doing a crossword puzzle with data. It's really kind of entertaining for somebody like me, but actually extremely frustrating. And the top line result is very similar in both cases, actually. About a third of the papers are fully reproducible, meaning you can get all the numbers from the open data without contacting the author, only a third. Then, if you reach out to the authors, and authors tended here to be very responsive because we had put in so much work already before we wrote to them. So nearly all of them wrote back and tried to help us, and only about 2/3 of the total were reproducible, even with author assistance. That means in a full third, neither we nor the authors could figure out the provenance of at least some of the numbers in their results section for one of their studies. So I found this very distressing, and it suggests further that open data policies are a good start, but they're not enough. Okay, so these are all general psychology-wide findings, but in the developmental context, we have these concerns and more. So for example, there's broad lab-to-lab variability. There are methodological differences. There's population variability. There's all kinds of superstitions around moderators of developmental findings, like blue walls make the babies depressed or cry or whatever and not do your study. Men with beards like me may make babies cry. I think that one's true. Second session babies are anathema for some labs. You can't test in 2 experiments at once. Other labs test all their babies in the second session. And they're always getting the babies that are passed on by other labs. So there's tons of these kinds of hypotheses. Furthermore, in development, there's little direct replication because data collection costs so much. And also because often data collection relies on a very specific procedure that you've implemented in your lab and that's going to be very costly and difficult to implement in another lab. Maybe you need a perfectly featureless room. Maybe you need a head-turn preference procedure set up, and so forth. Finally, there has been a limited tradition of data sharing and analytic reproducibility in development, in part because of the limited amount of data. It's felt like it was unnecessary to worry about the complex pipeline. But as our studies have shown, even very simple data processing pipelines are subject to error. Okay, so how do we respond now that we know that things maybe aren't fine, that there are real tweaks that we can make to our scientific ecosystem to try to promote better outcomes? Well, first, my primary engagement with methodological and substantive reform in science comes through the lens of open scholarship. I think it's really critical to think about a response to issues of reproducibility and replicability as transparency, openness, and sharing. And this has many, many facets, and I won't talk about all of these today, but I want to highlight that nearly everything we can do to broaden and open the scientific infrastructure helps with our shared goals of verifiability and transparency. So if I think about the open science movement, I think about it as a set of beliefs and research practices and results and policies that are organized around the central role of transparency and verifiability. And that critically, that means transparency and verifiability for everyone, as opposed to just for a select subset. So the more that we can open, democratize, and make equitable our science, the more we can actually have the requisite number of eyes on the critical problems that we want to address. So I think there's really a lot of room here for transparency and openness reforms to make science work better. And I'll just point to some work that I and others have done to try to make transparency and openness policies kind of accessible to folks. This is a guide that we published a couple years ago that has, I think, a lot of advice around how to begin the process of adopting transparency policies in your own work. But these are all individual level solutions, and they're largely, I think, solutions to the issues of reproducibility and maybe to a certain extent replicability. So what I want to turn to in the second part of this talk is thinking about models for cumulativity specifically, not just making sure that individual findings are reliable, but trying to move beyond them to a broader picture of human development and of the kinds of theories that we want to build. So when you go to a textbook or a review article, even very good review articles like this one by Pat Kuhl, which I'm using as a bit of a straw man here, you often see a summary of development that looks a bit like ages and stages. This phenomenon or this behavior emerges at this time period. And if you want to think about this from a theory building perspective, it has quite a number of issues. We point to a binary point of emergence. We think about stage-like transitions. There's a lack of variability in the estimates. There's no cross-cultural or really cross-individual variability noted. So if we had a particular milestone, what might our more cumulative model look like? Well, maybe we'd want to actually have a quantitative measure of the phenomenon, effect size or otherwise. We might want to supplement our estimate by some scientific uncertainty. We might want to map a developmental trajectory, and we might want to think about variability in that trajectory across populations or individuals. Then we might even want to link that trajectory to other phenomena that are posited to be a reflection of the same construct. Now, this is a little bit pie in the sky, but that vision is something that we've been trying to move slowly towards in a variety of ways. So let me tell you about those three ways that we've thought about this. The first is through synthesis via meta-analysis. And so my collaborators and I have been working now for a number of years on the site MetaLab, which synthesizes and brings together meta-analyses from a variety of different domains of development. We scrape these sometimes from the literature. We have challenges to ask folks to contribute their data, and we try to get these data at an open, standardized format so they can be manipulated and downloaded by anyone. This project is led by Christina Bergman and Sho Suji. There's an advisory board of folks that have been working on this, trying to engineer the site and get contributions. Just one insight from analyzing these data, not from a theory building perspective, but from a methods perspective. If you look at the power that's achieved, the statistical power that's achieved by most of the studies in these meta-analyses, it's really quite low. And so one basic recommendation that we made here is that if you start with n equals 16, n equals 18, the medians in the literature, this is just simply not enough. What you need to do is think about the phenomenon that you are measuring, calculate your power prospectively, and then try to actually do a study that's well-powered enough to increase the evidence base in whatever that phenomenon is that you're interested in. Happy to say more about this, but just in the interest of time, I want to show you a little bit about a couple of other strategies here. So the second is data aggregation. So meta-analysis is a very specific type of aggregation across empirical findings in the literature, but there are some cases where standardized methods or instruments make it advantageous to aggregate specific data from that particular kind. And luckily we have very good inspiration in the field of child language. So the Child Language Data Exchange System has been an open data project for almost as long as the field has been a real community. So Brian McWhinney, Catherine Snow, and other early pioneers here have been sharing data openly and transparently and reproducibly for many years, and that's a very exciting model. And then the MacArthur-Bates Communicative Development Inventory, which is a standardized vocabulary checklist, provides another kind of model for open sharing of materials and the normative data and so forth. So inspired by these predecessors in the child language field, we created a site called Wordbank. Wordbank takes questionnaire data from not just the MacArthur CDI in English, but the dozens of adaptations of the CDI to other languages and aggregates them again in an open and transparent framework that allows for their download programmatically through R and also through their manipulation in an online interface. So the idea here is that you can really test your own hypotheses in this ecosystem. You know, Sam has used this, others have used this to try to make kind of more cumulative claims, more cumulative theories on the basis of the broader evidence base for the acquisition of words across many different languages. So just to give you a flavor for what this could look like, you can browse around the data. These are English acquisition data of individual children's vocabulary across age. It's cross-sectional data, but we could look at German. We could choose a language. We might split this by any kind of different variable, like in this case, gender or sex. We could take the medians and recover a female advantage here. And now it's relatively easy to test that in different languages. So here it is in Korean. This is the female advantage. We could see whether this shows up in, say, Swedish, which it does. So this kind of larger data set arranged appropriately gives you a tool for exploration and really kind of testing out theories, which can then be implemented in experiments or one-off designs that then kind of confirm a conclusion that you found in these original data. So the goal here then is to try to make our evidence base more widely shared and allow that to be used more transparently by the entire field. Okay, so those are two projects on synthesis and on data aggregation that try to create a cumulative evidence base for the field, but there are cases where the data that we want are not out there. And so for that purpose, I'll turn to my last kind of suggestion or thought, which is going to take up the remainder of the talk, which is that sometimes what we need to do is to collaborate. And so, as Sam alluded to in his introduction, we decided to think about what a large-scale distributed collaboration might look like in developmental psychology. And it's a collaboration we, tongue-in-cheek at the time, but now seriously call ManyBabies. So, in order to explain the ManyBabies collaboration, let me start at the beginning and just tell you about our very first ManyBabies study before we knew that we would have a consortium and a governing board and a whole infrastructure for trying to create these collaborations more continuously and more consistently. At the beginning, we were just a bunch of folks chatting about what this kind of collaboration might look like and where you might want to start. And we all agreed that you'd want to start with a phenomenon that's important, and also a phenomenon that maybe is replicable, that we all think is a real phenomenon, so that we could be trying to understand not just is a phenomenon replicable and real, but also how it varies across labs and individuals. And of course, you can't look at variation in some phenomenon if it doesn't replicate at all, if everybody just gets zero effects. So we tried to start with something that we thought was interesting, but also a relatively conservative choice. And for that, we turned to infant-directed speech. So I think maybe most people in the audience will be familiar with this, but in case you haven't encountered infant-directed speech, it describes the phenomenon that adults talk with a relatively monotone pitch or relatively low variance pitch when they're talking to other adults. So this plot shows the fundamental frequency of the pitch in a sentence. So this is a mom whose voice is higher than mine, but she's saying, "I had a little bit and the doctor gave me Bendictin for it." So pitch is changing just a little bit, but it conveys a real signal, but it's not very variable. Then that same mom is talking to her baby and she says, "Can you say ah? Say ah. Hey you. Say hi. Hi." So she's crossing many octaves in her pitch. This pitch variability is very characteristic of infant-directed speech, where you also get shorter utterances, more repetition, longer pauses, a bunch of stuff that seems like it might be beneficial for language development, both by drawing children's attention and also potentially by creating a simplified, maybe rationalized signal with a bit more learning potential to it. Okay, so the phenomenon we selected to look at was the infant-directed speech preference, because it turns out that babies like infant-directed speech. A classic paradigm for studying this is visual preference. You show a checkerboard like the one that's shown here, and you play infant-directed speech or adult-directed speech, and you see if the babies look at the checkerboard more when it's accompanied by infant-directed speech. And you alternate pairs of infant-directed or adult-directed speech trials. And there are many findings of this sort in the literature, so that made it this perfect for a first collaborative replication. So what we did in this project was create infant-directed and adult-directed speech clips. We got these from naturalistic observations of moms in the lab with their babies. So here's what the IDS sounded like.
Eating the shoes. You want to grab that part? Here we go. Do you want to hold the handle? Look at that. What do you think? Can we roll the ball?
Yeah, you can put it on your head like a hat. It's got white stars. There's 50 of them.
So it sounds pretty normal. It's people talking to kids. But here's the adult-directed speech.
Slightly bigger, but squishy in the same way. It's just a blue kind of basketball.
So it's good for mixing.
Reflects the light pretty nice. It has little divots in it.
With trucks all over it and then Velcro.
So you could see the adult-directed speech has a different register, shorter, lower pitch, less variable, just precisely the way I was describing. And we decided at the beginning, rather than make this a strict replication in every lab being the same, we actually pushed this out for a variety of different procedures. Some labs had babies look at that central checkerboard fixation. Others had an automated eye tracker tracking the child's eyes. Still others used what's called the head turn preference procedure, where they didn't use the checkerboard, but they had the child turn their head to different sides. And we developed these stimuli, we developed the collaboration, and then we submitted a registered report, got the protocol approved, and pushed it out to labs around the world. And we were just overwhelmed by the amount of interest we got. We heard from 68 labs across 17 countries, 4 continents. We ended up with 2,700 babies in our initial data set and retained about 80% of those for 2,200 babies. So this is really, I think, the largest experimental study of infant language that I'm aware of. And this took forever. Let's just be honest. Collaboration is slow and complicated. The original idea came around in maybe March of 2015. The call for participation was in December. We decided on infant-directed speech shortly thereafter. Then 2017, we got the stage 1 accepted and began data collection. We presented the first results a little more than a year later at ICUS. A year later after that, the stage 2 registered report was accepted, and the paper was officially published after endless typesetting and formula checking at AMPS in 2020. So it was really quite a journey. But the data were really, I think, very interesting and worth it at the end. So here's just a sense of what we got. So this is a meta-analysis, not of different findings in the literature, but just of the different labs contributing data to our sample. Some labs contributed data at different ages, some at only one age. Here it's broken down by methods, head turn preference, eye tracking, and central fixation. So the overall meta-analytic mean here was 0.35. So that was significantly above 0, but it was about a third of a standard deviation. Not a huge effect, but a real effect. It was substantially moderated by method. So central fixation yielded about 0.29, eye tracking 0.23, and head turn preference, surprisingly this older, maybe a little bit more complex paradigm, yielded a larger effect size quite reliably. Now, even head turn preference didn't match the effect size from the literature, which was closer to 2/3 of a standard deviation. And we've been really thinking about why we see that gap. And it's not explained by any of the moderators that we have in our data set. So we've got a paper coming along on that question. But maybe most strikingly for me, there was no evidence in this meta-analysis for what's called heterogeneity, the idea that there are moderators beyond sampling variation. So although the literature has focused on the idea that small moderators could make a big effect to infants' behavior, what we found here was not consistent with that. In fact, what we found was more consistent with the idea that small samples lead to big variability, and that there is a real infant-directed speech effect. It may vary modestly with some of the moderators that we were interested in, but the primary reason why these estimates from the meta-analysis are all over the place is simply that the samples for any individual lab were small. Now, one of the things that we didn't anticipate at the time of beginning this project was just how much would come out of doing this single project, how many spin-offs and other potential projects could emerge. So one thing that was very exciting was the emergence of the ManyBabies 1 bilingual spin-off. Bilingualism is, of course, a huge topic. Most children in the world grow up with more than one language, and despite that, developmental and language research tends to be biased towards monolingual populations. Also, bilinguals are harder to recruit on average, especially in the places where many of our labs are based. And so the ManyBabies approach here of collaborating to get larger samples was really important. So the bilingual spinoff was led by Krista Byers-Heinlein and kind of proceeded in parallel. The idea in 2016, the stage 1 and data collection kind of concurrent with the original project, and then the stage 2 submitted in February of 2020 and published just this year, actually. And just one finding from this pretty exciting paper. Overall, we found the infant-directed speech preference no problem. It was really quite clearly there. But we also did find that it was clearly moderated by North American English exposure. So the stronger the infant-directed speech preference, the more exposure the child had to in their particular home and environment to North American English, the stronger their preference was. And so that did suggest that bilingual exposure was being measured appropriately and that it was moderating the size of the infant-directed speech preference, even though bilinguals do show the same, overall in the average, the same preference. And this is just one of many spin-off projects from MB1. We had a gaze following project that was published in AMPS, a test-retest reliability paper that's currently in paper writing. Unfortunately, reliability of this measure is quite limited unless you have lots of trials. So only a few babies contributed the maximal number of trials, and those are the only ones that have reliable measures for them. We've tried following up with longitudinally with the CDI. Didn't really relate to the infants, probably because of the reliability issue. We've compared to the meta-analysis and are working on this, so it's a comparison between multi-lab replication and meta-analysis, trying to understand publication bias and the way the literature works. We have an extension now in progress, extending to Africa, which was not a continent that was represented in the original data collection, and we've just had our first babies tested in Ghana. That's very exciting. And then there's a project re-recording the stimuli across languages, which is going to assess both IDS implementation in the languages and language effects. So just a tremendous amount going on here. And ManyBabies now, because of the success of this project, I think has grown really remarkably. So we started ManyBabies in 2015. By 2016, we had begun a theory of mind project, ManyBabies 2. MB4 for social evaluation started in 2017. MB3 for rule learning started in 2018. By 2020, we had a study on infants' attention, an online testing project, a demographic project, and now this year we're starting a network of networks, really trying to take this kind of big team science concept more broadly and formalize it and try to create communities around it. So I'll just very briefly mention the topics of these other projects. The MB2 is a theory of mind and infancy. We're trying to build on anticipatory looking paradigms to really test this very tricky question. I can't help but play you the stimulus because it took years to make and I love it. So the idea is this is a paradigm about tracking beliefs. So we've got bear and mouse, and they're playing a game. Here comes mouse in the tunnel. We should anticipate with our eyes if we know where he's going to go. Oh, he went over there. Bear sees him. Now Bear's going to come find him. Where's Bear going to come out? Feel your eyes pulled toward the correct target. So that's the general idea. We're going to use this method to try to initially just track belief tracking at all, and then false belief in a follow-up. ManyBabies 3 actually circles back to this rule learning phenomenon that I was looking at in this initial multimodal rules paper that I mentioned at the beginning. And of course, that's been a topic since far before that. ManyBabies 4 looks at the Hill-Show paradigm of Kylie Hamlin, the helper-hinderer paradigm that's been controversial but also really important for a lot of theorizing about early social and moral judgments. And ManyBabies 5 looks at changes in attention and the puzzle of novelty and familiarity preferences in infant preference studies. Really trying to use the scale that we have to address this broader theoretical question. So ManyBabies is a broad and growing community. We really invite you to learn more and to join us. There's a lot of information on our website. Just go to manybabies.stanford.edu, and you can learn more about the projects that I talked about and many more spin-offs and nascent projects. Just to highlight a little bit of what I think is unique here, ManyBabies brings together key theoretical tests and methodological innovation and improvement, really trying to put together methodological best practices with replication and theory in a single package, try collaboratively. We have consensus-based designs. We study methodological differences, trying to help labs make rational choices about what they do with their methods. We also create lots and lots of spin-off projects, trying to create high-value data sets that answer more questions and provide opportunities for many more people to lead, including early career researchers. Our model is fundamentally consensus-based. We try to let people explore open science with a safety net as part of a network and community that can discuss and teach, as well as just kind of pushing out things into the broader community. And finally, I think really what's different than many other Metascience projects is that this is a radically collaborative framework. The goal is just to have a community that's working together to better our science. Okay, so just to wrap up now, I think that it's clear that my initial errors and missteps are not unique, that problems of reproducibility and replicability are substantial, and they are substantial challenges for behavioral science and psychology. And there are many steps that individual investigators can take to improve their practices through adopting transparency reforms. But more broadly, I don't think it's just about correcting practices. Correcting practices and changing our workflows is important. But there are also ways that we can engage in, I think, creative and fun new ways to look at data and look at the questions that we care about that can drive our science forward. And so thinking about meta-analysis, data aggregation, and collaboration as ways of increasing our power and precision. More broadly, these kind of incremental changes, I think, can bring us closer to a cumulative research practice that really helps us build a stable and robust science of child development. Thanks so much.
Great talk. Thank you. I can see we already have one question in here. So this is from Prana Aneja, who's a wonderful PhD student at UEA in Norwich. Has ManyBabies considered collaborating with not-for-profits in more lower and middle income countries? I imagine not many labs in those countries are signing up yet, so reaching out to not-for-profits could be useful.
Hi, Prana. Yeah, thank you for that question. Absolutely. So ManyBabies began as a collaboration of convenience for labs that were interested in these kinds of questions, but we've quickly realized the critical importance of having a more global footprint and really trying to create a network that allows us to study infant development across the world and also allows us to provide mentorship and provide a network for folks in low and middle income countries that are interested in these questions, that are interested in kind of participating in bigger and broader projects. And so we're really actively trying to understand the best strategy for pursuing that goal. So we've done this in a couple of ways, and we're eager for the involvement of folks that have connections in lower and middle income countries and can think about the right stakeholders to engage with. So one of the projects that I've been more deeply involved with is ManyBabies Africa, where we put out an open call, really tried to find connections and pursue those for folks that would be interested in participating in a ManyBabies study. And then we convened those folks in a conference, did training, provided equipment, did a research exchange to learn about the topics that they were interested in, and then wrote together our registered report, which is, now has data collection ongoing. But that's just one particular model, and something that we are really enthusiastic about is also thinking about ways to collaborate on questions that are generated in other contexts. So thinking about having a process for the kind of organic creation of ManyBabies projects from a bunch of different locations and a bunch of different perspectives, and that's something that we're really working on right now.
Question here from Durham's very own wonderful developmentalist, Dorothy Cowie. Do you relate to the Brewery project at all? I've come across it from following Karen Adolph's work. Thanks for a great talk.
Hey, thanks, Dorothy. I appreciate it. Yeah, so Brewery, for those that don't know, it is a wonderful project provided by Rick Gilmore and Karen Adolph. It's a video sharing site hosted by them. And one of the key things that's sort of special about it as a video sharing site is that it solves some privacy issues by virtue of having a fairly restrictive and complex licensing process for the users of the data and a corresponding consent process for the posting of the data. So it allows us to post more developmental data and ensure parents that those data are only being used for appropriate research. So my lab uses it fairly extensively, actually, for some of our work with head-mounted cameras. We share corpora like the SayCam corpus on Brewery. And ManyBabies is also interfaced with Brewery a fair amount. We're actually doing a CDS pre-conference with them in Madison next spring, if folks are interested. One of the uses that we've made for it that was really fun was the posting of walkthrough videos for each lab doing the ManyBabies 1 procedure. And so you can go on Brewery and use these for teaching or for learning about the different procedures that baby labs use if you're thinking about setting up or modifying your own.
Brilliant, thank you. While some others maybe think of some questions, I had one for you. You started off with quite an excellent demonstration of, what can go wrong with some studies, particularly, as you're learning how to both run studies and how to sort of engage with them, engage with the analysis as a student. We've obviously got a number of students in the audience. I wonder if you can say a little bit about, what your recommendations might be, for students who are starting out, probably running their first student projects and things, where to start to avoid some of these issues, or indeed, to dive right in or something.
Yeah, this is a great question. And so I actually am teaching my favorite course right now, which is experimental methods for graduate students. And one of the key failure modes for that class is that students hear me recite this litany of mistakes and horrors and think, how should I even get started? Aren't there just, so many mistakes to be made here? Is everything kind of weighted, and is there are pitfalls at every corner? And I think my message, which we hopefully take in the practical guide paper that I recommended here, is that you have to start somewhere. You have to begin and begin with the practices that you're most interested in and the kinds of transparency moves that you think are most comfortable within your own workflow and your own context and lab. There isn't open science and transparency is not about kind of purity or about kind of achieving some ideal. It's about kind of concrete and practical steps along a path towards increasing the value of your own work via these kinds of transparency moves. And every step that you take can increase your value. So if you have concerns about posting data because of privacy or restrictions, post code instead, post materials. So if you are, worrying about kind of access and open access issues, consider posting preprints that will help you get your ideas out early and increase access without that kind of extra cost. So the idea is to take incremental steps that increase the benefit and transparency of your work. And then think in combination, often with supervisors, with others in your program, about the ways that these practices integrate with the kind of science that you want to do. Because one of the key, I think, take-homes from ManyBabies is that there are different ways that open practices can interface with different kinds of science and different kinds of projects. That there's not one size fits all type of approach.
Thank you. I couldn't agree more. We've got a question here from Lily. Thank you for the inspiring talk. Do you know of any cumulative science projects with older children? Is this in the pipeline for the ManyBabies team?
Yeah, great. So fundamentally, we will consider proposals for new projects whenever anyone wants to bring forward a new project. So if you have a particular experiment or theoretical construct in mind, we'd love to hear from you. We can help match you with others with similar interests or consider a proposal and provide feedback. So there's very much a goal of trying to promote projects in the developmental space. With respect to specific projects that are ongoing, one that I know of is the Many Numbers collaboration, which is aimed at numerical cognition and development in early and middle childhood. So it's kind of more preschoolers. I think there's an infancy wing, but it's mostly kind of preschoolers to early elementary. And I think this kind of collaboration, if anything, could be easier with kind of preschool children who are often easier to test than infants and where the methods can be more standardized. So that's certainly a direction that I'd be interested in going in.
Thank you. One from Taiwo Bello. In your research studies, do you include children with disabilities such as autistic and ADHD? And what developmental strategies will be appropriate for them?
Taiwo, thanks. Yeah, that's a great question. So for the most part, we don't see diagnoses for those particular developmental disorders as early as the studies that I was telling you about. Right. So most, autism spectrum diagnoses are now moving earlier, but the age of diagnosis is typically, minimum kind of 2-ish. And often substantially later than that. So we haven't engaged with diagnosed developmental disorders in ManyBabies. On the other hand, work with clinical populations is a very exciting frontier for collaboration. Because of the heterogeneity of clinical populations and the difficulty of collecting such samples, collaboration has been a method for clinical research for a long time. Often the model has been closer to the traditional multi-PI funded consortium grant, in part because of the greater costs there. The ManyBabies model substitutes that relatively more traditional model for a more flexible model where we try to spin up collaborations slightly more quickly and more organically. The downside is that kind of really costly work with very tough populations to recruit that require a lot of extra specialized expertise for assessment and diagnosis, that could be complicated. So we haven't had a project on clinical populations yet, but I think the consortium would certainly entertain one if somebody wanted to lead one and bring it to us.
Excellent, thank you. I'm just aware that it's sort of 5 o'clock. I think there's just a couple more questions. If you're okay to stay on, we can just answer those. And if people need to leave, thank you for coming. So we have another question from Prana. In regards to the slide about the power, where you mentioned that single labs often have lower power, would it help if labs increase the number of trials, considering that recruiting infants is not easy? And I guess this is an interesting question, particularly in the context of some of the paradigms that we have, which are, historically, I guess, published with sort of 6 trials or something. Yeah,
that's a fabulous question. And one of the things that I highlight in talking about power and effect size in my class is that fundamentally, effect size is relative to variability. And same thing for reliability. Reliability isn't absolute in the ways we compute it. It's relative to variability. And so if you can do things to reduce variability, you can increase effect size and hence increase power. And that's absolutely the strategy that's been taken in adult cognitive psychology research, psychophysics, and so forth. They have very low ends, but very high reliability because of so many trials. So I think that's certainly a great strategy. There's a paper by, I think if it's Michaela DeBolt and Lisa Oakes that investigates this quantitatively and suggests that due to the variability in infancy paradigms, you can actually add trials or add babies, and they do kind of relatively equal things to the power and reliability of the paradigm. For me, the challenge is always thinking about how those extra trials can be accommodated in the particular paradigm we're interested in. So because of the nature of babies and habituation, sometimes you can't sandwich in too many more trials, but maybe you can do another session with the same babies. That's another solution. We've tried to push this pretty far in the preschooler realm. So some work that Abdella Fortasi and I did. We did this kind of very psychophysical 60 trial experiment with 2x2 design within subjects for preschoolers. And it was really fun to try this out. We had a truly heroic research assistant, Sophie Reagan, who's now a PhD student working on this kind of stuff. Who shepherded kids through these 60 trials, painful 60 trials of phoneme discrimination and word learning and so forth. But the trade-off is that your retention rate is much lower. Perna, now that things are moving online, we could do. It's much easier to do several sessions. That's absolutely right. Especially in unmoderated platforms like Lookit, where it really is kind of possible to push things out and have. Multiple sessions. I mean, there's still obstacles, but that's absolutely right. And a number of groups and consortia are pursuing large Lookit-based batteries where you try to increase reliability by multiple sessions. So that's definitely a next frontier. It would be exciting to see where that goes in the next couple of years.
Great, thank you. And then I guess, one final question from me is, you showed the map of studies that you had for ManyBabies. And I mean, I know you've mentioned the ManyBabies Africa project, but what can we do? Are there any steps that we can take that you already have in mind to make representative more generalizable to sort of the global population?
Yeah, so I think that the key step is creating networks of folks who are engaged with the community and the scientific questions and trying to collaborate to have these kind of local networks that are outside of the usual suspects, the US, Canada, Western Europe, and who are interested in developing projects together. So just a kind of admiration and shout out to the folks at LSCP and at the Max Planck Institute, so Alex Christia and Kara Rowland, who've really been working with the L+ Summer School for Language Acquisition. It's a global summer school, and the Langview Consortium to really try to bring folks from lower and middle income countries into the international community around language development and to have a conversation about what that might look like.
Brilliant, thank you. So if there are no other questions, I'd like to thank you so much for what was, for me, at least an absolutely inspiring talk. It was brilliant to hear about, both past mistakes and, future directions and exciting current sort of multi-lab projects. So that was absolutely brilliant. Thank you.
Well, thanks so much for having me.